So we just took turns writing the resolution. So we just like asked him, and what do you want to do? I've done it twice. We just started that series where
All right, let's get started. It's after 2 o'clock. My name's Kevin Miklash. I work with Iridescent. It's a nonprofit dedicated to educating and inspiring the next generation of students. I'm actually Iridescent's most recent hire. I started working yesterday. But I'm actually quite familiar with them. I've been volunteering with them for the last 4 years as a graduate student, as a scientist, so I'm quite familiar with how they operate. What I'm going to tell you about today is more of a theme than an actual project that I've been working out with Iridescent over the last year and a half, which is thinking about cooking as an analogy to the scientific method. In particular, the way that you would generate a new recipe and come up with the component to that is very similar to the way you come up with new knowledge in using science. And so I've kind of developed this analogy and pursued it in a couple different ways. And so I'm going to tell you about how we've used this analogy and how we've developed it. A little bit more about me. My background is mostly as a scientist at this point. I got my undergraduate degree in physics, and my PhD I just finished this month in biology. And I've actually done a fair bit of engineering work along the way too, so I've kind of dabbled in a couple different scientific fields. And I've been really interested lately in what are the commonalities between those different fields, how are different types of science really related and similar. And so that's what I'm really going to talk about today. This is going to proceed in 2 sections. In the first section, I want to talk a bit about the scientific method, what we usually think of it, what are some new interpretations of it, and what's the interpretation we've been using with Iridescent in this project. And second of all, I want to tell you about the few ways that we've implemented it so far. And I'm really looking for more feedback on how we can use this analogy and do more with it. So I'm hoping to get some really good ideas out of this. So I wanted to start with a quick video kind of showing a little bit more about what Iridescent is about. As I said, I got involved with them as a scientist. Iridescent really tries to be the bridge between researchers and universities and schools and students. Finding ways to take concepts that researchers actually use and find clear language and good analogies to put it in a way that even a 4th grader can understand. And so this video is sort of geared towards that. I don't have speakers, but I have a microphone, so let's see how this works. Everyone hear that?
Everyone has something that they're running from. One comes to a close just as another one's begun. Days turn into nights and nights to years, yeah, time goes on. Oh, and everyone is trying to find a place where they belong.
What's wrong with this one? This is pretty cool.
It is pretty cool, actually. This is what we call an airfoil. You probably know it as a wing. As I grow older and live, that there's more that's waiting for me. More I have to give. Oh, I've tried for way too long to work for something good when it was free, waiting for me. But I didn't take it till I knew that I could. Yeah, I can finally see as I grow older and live that there's more waiting for me.
So as you can see, this video is more targeted at the scientists, getting them interested in working with us. And so, like I said, they clearly hooked me. And so what I want to talk to you about today is one of the things that I've gotten really hooked about. So let's talk about the scientific method. First thing I want to do is ask you what usually comes to mind when you think about the scientific method. What words or ideas or adjectives do you usually think of when you hear the scientific method?
ask a question.
Ask a question, so asking questions.
Experimenting.
Asking questions. Experimenting. That's another good one.
Hypothesis.
What was that?
Hypothesis.
Hypothesis.
Measurement.
What was that?
Measurement.
Measurements.
Observe.
Observe. Thanks, someone back there. Observations.
Data.
Observations. So these are all sort of things you do with the scientific method. What sort of things what are maybe adjectives you associate with that? What sorts of connotations the process has?
Objective.
Objective.
Systematic.
Objective, systematic. I don't know, space.
Trials.
Testable.
Procedures, steps.
Procedures, doing it in like testable order.
Linear. Some students think it's linear.
Linear. So you generate new ideas with science too, right? Does anyone associate the word creative with science?
Innovation.
Innovation, creative. Those ideas can be associated with it too. I'll put those down because I like those. So this is a pretty good list.
Try.
So what's really interesting about this list is if you look through it, there's really two different sorts of ideas about the process of science that are really being offered here. First of all, you've got all these linear procedures, systematic, objective. These are very rigorous, germane ideas of the view of science. But asking questions, thinking about things, being creative and innovative is a very different view of science. And how can this process be both very rigorous, very structured, and then also very open-ended? Is really sort of a tense interplay in my mind. And how can one process embody both of those things? And that's one idea I want you to keep in mind as I kind of go through this, and I'll come back to this point. So I wanted to show next was some models of the scientific method. And I pulled this one up because I think this was probably pretty much the model I saw when I first learned about this in fourth grade. It was very similar. And so I don't want you to pay attention to the words here. I just want you to look at the structure of this model. And what does the structure of this model tell you or imply about the process of science? Just the way it's structured, what things can you say?
One way.
What was that?
One way.
One way. So there's one way to do it. If you're not doing it that way, you're not doing it correct.
Well, it's very orderly, first do this, then this, then this, then this, and there's not any flow back.
Exactly.
Maybe you might revise and go back through again.
It's a step-by-step process. You have to do it in order, and you can't redo any steps.
Yeah.
If you've done it wrong, you're supposedly screwed or something. What else does this shape tell you?
Linear.
It's linear. What do lines have? A beginning and an end. So what does that imply? Line segments.
Line segment.
So line segments have a beginning and an end. And what does that imply? To start and a finish. And once you've got to the finish, you must have...
It must be done.
You must be done. You must have gotten an answer. So so when you finish the scientific process, you should be done. You should have an answer. And if you don't, you've done it wrong. These are all things that are implied just by this structure. It doesn't really matter what's even in the boxes here, what words are there. Students will form these ideas just by looking at this general shape. And so we really have to ask ourselves, are these the implications we want students to pick up about this process? Now this is a newer view of the scientific method developed by a couple people at the University of California, Berkeley. You can find it at understandingscience.org. I really like this. They developed this by interviewing actual scientists about their research and mapping out different steps of the scientific process that they went through in actual experiments. And then they basically made a flow chart that mapped that process and relayed all the different connections, pathways they took. Now looking at this, you can see it doesn't matter what the words are, you get a very different impression of the scientific process. There's a lot of feedback loops. You can zigzag around either from circle to circle, or even within circles, there's a lot of feedback that can occur. So this is a very iterative process. There's no clear endpoint here, and in fact, wherever you are, you can keep going somewhere else. And there's not a clear beginning, although at the top there is an input there, but you can actually start this process from anywhere in this flowchart. So this gives a very different view of the scientific process. And what's cool about this, since they developed it by interviewing scientists, they can actually map a research experiment on this flowchart. And so that's what they did here. What I'm going to show you is Dr. Alvarez discovered the asteroids killed dinosaurs. Okay? And so this seems like a pretty simple discovery. So what I'm going to show you is basically walking through the steps that he went through when he made this discovery. And this is again available on their website. So starts up at the top. You see immediately there's a lot of feedback. He's going through parts of the process again and again. He's repeating steps. There's a lot of loops going on. He's revisiting parts that he's already been to, apparently reforming ideas or something like that. And you can see there's one part of this process, this part right here, that's about to end, that he goes through several times in the same order. And so, what's going on there? Why does he keep going through that part? Well, if we go back to this and zoom in at the middle here, this is generating a hypothesis, getting expected results, and then comparing them to your actual results, and then doing something with it. This is basically doing an experiment. That's that linear process of science. And he's going through that process in the order described by the scientific method. But that's embodied in this whole other structure. There's a lot of things that lead into that, and a lot of different ways you can lead out of that. So what this really does is it embodies that original linear process, but it encompasses it in this much more iterative feedback framework. So that was clearly a very complicated sort of view, and so when I was trying to think about what conception of the scientific method I wanted to promote, I wanted to start a little simpler than that. And so I went back to this linear scientific process, and it's basically question, hypothesis, do an experiment, get results, put them in context. I wanted to take this and add a little bit more to it, make it a little more realistic. And so, well, the first thing I did was I took out the hypothesis step. Hypotheses are a pet peeve of mine. It's my chance for mini rants against them. Basically, I could go on about hypotheses for a while, and if you want to hear it, come up to me afterwards. But the short end of the story is that hypotheses are not used the same in every science. A lot of scientists use hypotheses very differently. And even in those that use them, a hypothesis is basically just a predicted answer to a question. And if your question is well formed, I think that the hypothesis is so obvious as to not even be helpful to the experiment. And so really all the focus should be on the question, developing a good question, and not on the hypothesis and expect an answer to this question. And so I've really taken out this step and haven't considered it very much. And you just basically generate a question, answer it with an experiment, look at your results and put them in context. So this is the basic process I've looked at, but as we know, there's a lot of things that feed into this. Before you come up with your question, there's a lot of things you can do after you've gotten your results. So I'm going to add a couple arrows to kind of take into account of that, and I'm going to color code everything. So as I talked about in the beginning, there were two conceptions of science. One is this creative, innovative process. The other one, I'm going to put boring here, but that would be that rigorous, objective, monotonous process. And as you can see, it actually has both of these components in it. They just occupy different parts of this process. So coming up with questions and developing a question is actually a very creative endeavor. It's not something that just happens. There's lots of ways you can come to it. It can either be from the results of a previous experiment or from some ideas you've gotten in some other fashion. Turning that question into an experiment is also very creative. Often you have to invent your own methods or methodology. I mean, there's classic, control and treatments that you can do, but that doesn't always work. And sometimes you have to be very creative in how you do an experiment. Once you've got your experiment set up, though, collecting your data, analyzing the results, doing statistics on it, whatever you're going to do, there are very set ways you can do that. And it is important that you do these in a very objective, rigorous way, because that allows you to firmly answer your question in a very objective manner. So this is an important part of the process here. But once you've gotten your results, figuring out what you want to do with them, what context you want to put in, what have you added to the knowledge base that we have of science, and what further questions can you ask based on your results? These are also very creative things that require innovative thinking. And so what I've decided to do here is I've decided to focus on basically all the orange parts of this scientific method in this process, which are the ones I think that haven't really received a lot of focus previously. So how are questions formed? How do you transform a question into an experiment? And what do you do with the results? And so question then is, well, why would you use this model? Why would you use this conception of a scientific process and focusing on the orange parts of this process? And basically, I have 3 reasons why you should do this. First of all, it's more engaging. These are the exciting parts of the process, the creative parts of the process. And so all other things being equal, why not focus on the parts that are more exciting? Second of all, it's more accurate. These are the parts where, as a scientist, you spend a lot of time in these parts. I mean, you spend some time plugging through an experiment, but you spend a lot of time thinking about the experiment and figuring out what to do with the results. And so focusing more on those parts is a very important part of the process for science, and it's almost more accurate to put a lot of emphasis on those when teaching the scientific process. And third, it emphasizes qualities of good scientists. And what I mean by that is, I got interested in this idea of, I guess, philosophy of science pretty early on in grad school. Since I was coming from physics to biology and doing engineering. When I would go to conferences, I would ask people, what scientists do you really like, and why do you think they're a good scientist? And the answer I got more often than anything else was, I think so-and-so is a great scientist. He knows how to ask just the right questions. His questions are always so insightful. 9 times out of 10, it was this part of the process right here that distinguished a good scientist from a great scientist in the minds of other scientists. I mean, occasionally you would hear someone that would say, oh, he's such a great scientist, he's such a meticulous experimenter. But that was pretty rare. And so, why not focus on this thing that is recognized by scientists to be the distinguishing feature of what makes someone a really great scientist? So, with that, I'm going to switch gears and start talking about how we've implemented this sort of method, but I want to stop for a second and open up to any questions or comments anyone may have before I move on. So does anyone have any questions at this point? Okay. I guess we'll keep going on and see how we use this. So there's 2 main ways that we've tried implementing this model so far. So cooking as science, 2 implementations. And so I want to talk about them one by one. The first way was in Top Chef Masters. I don't know if any of you follow this show, but in the last season they had a science-themed episode. Iridescent was actually contacted to be one of the coordinators of the scientific content on that show, and I was actually the lead science advisor on that project. I advised them on which scientific concepts to use and how to display those concepts, how the chef should be evaluated by the judges on what they did. I have a few more things to say about it, but first I'll play this little trailer video for you so you can kind of see what the episode was like and how it worked. Again, we didn't have complete control over this process. This was showed as clearly so, but it was still pretty interesting. So as you can see, they focused in on the viscosity, the scientists who covered that topic, and that was actually the one that I thought did the best job of doing an experiment showing the different viscosities, which was pretty cool. But as you saw, the judges were all chefs here. We had limited control over how this process went, what actually happened in it. And so I say I was satisfied with this process. I thought we did a pretty good job. And if you're interested, I'd encourage you to take a look at the episode and see how it was presented throughout the episode. I think it's available on Amazon. But we wanted a little more control over it the next time we did this. And so one more thing I want to mention about this, though, is you noticed in the video there were some beautiful posters of these scientific concepts. And this was one really nice tangible that did come out of this process. We actually advised the production of these posters with our graphic designer, Ilona, who's shown in the bottom corner here. And all these posters are actually available from her website if you're interested in any of them. Her name's spelled Ioana. And so these were really great illustrations of how these principles acted on both the macroscopic and microscopic scale. And this is sort of a little bit of a tangent from the using cooking as science. Yeah.
We would need you to spell the name again.
The name was Ioana.
I-O-A. Okay.
Ioana.
Okay.
And you can also find these on the website I'm going to talk about next.
Okay.
So these are really cool illustrations of what's happening on both the physical and chemical level, on macro scale and micro scale. Just to walk you through one of these quickly so you can see kind of what they're illustrating. This is the one I spent the most time doing, so I'm most proud of it. This is viscosity. The main part here is a graphic which shows how the viscosity across the top, as you get more and more viscous liquids, how the length of the drop that would come off a spoon as you poured these off would change. And as these liquids got stickier or more viscous, they would hold together more and create a longer and longer drop. Very clear illustration of the concept. What we also did is broke this down on the molecular level. So there's two main things that contribute to how sticky a liquid is, and that's the intermolecular bonds. How many of them you have and how strong they are. And so if you want to create something that's stickier, you either take all of your molecules that have these bonds between them and you pack them closer together so that there's more bonds per volume. Meaning when you try to move the liquid, there's more bonds that have to be broken and reformed. Or you just create a different kind of bond. The chemical bond is actually stronger so that when you try to pull it, it's more difficult to separate them and rearrange them. And so we really were able to explain this concept on both levels. And this was really fun to work through. But again, this didn't quite get at the cooking as science aspect that we really wanted to get at. So what we did is we developed, after the show, developed a blog called ScienceFare, found at sciencefare.org. This is the second project I'm going to tell you about. This was really fun. What we did was we went out and contacted other scientists, mostly grad students and faculty, and asked them to do experiments in their kitchen about topics that interest them, actual science experiments, and then to blog about it, write about it for a blog. And so what we ended up here was a bunch of stories from actual scientists, actual researchers, as they did the process of science. And we asked them when they did this to focus on those creative aspects of the scientific process that I mentioned to you earlier. How they came up with questions, how they transformed it into experiments, and what they did with the results that they got. And so we've been doing this for about 10 months now, and so we have about 24 stories on the blog, and we're still adding to it every week, whenever we can. The different ways that we've been using, or can use sciencefare.org, for example, it can be a good basis for classroom activities. Each blog entry is coupled with an activity or an experiment that can be replicated in either at the lab or in somebody's home. These also can make great in-class reading assignments, again, about the scientific method. The scientist is actually walking through the various steps of the method. And it could provide a good way to go over that. And also, this is a blog. You can post comments on it, and this is a way where people can actually engage with scientists about an experiment that they actually performed and ask them questions about why they did certain things. So what I wanted to do was tell you a couple of, basically walk you through a couple of the blog posts that we've done so you can get an idea of what the content looks like. So the first one I want to talk about was by Carolyn Teppel. Carolyn is actually the co-founder of the blog with me and a fantastic chef. She's another grad student that I went to school with. She's actually been asked to make 2 wedding cakes for friends, and a third one I think just asked her last week to make a wedding cake for them. Fantastic chef. She's also a geneticist, as you might guess here. And so she found herself on a research trip in Newfoundland, and she wanted to cook something, and she wanted to use something local, but there's not a lot to use in Newfoundland. So what there is a lot of, though, is berries. And so she went out and found 4 local species of berries and collected them. And she also wanted to learn about jam making, so it was a great chance for her to learn both things. But she knows that when you make jam, it's really important to consider how much pectin is in the berries. The pectin is the thing that makes the jam kind of form a nice gelatin. And so if a berry doesn't have enough pectin in it, you have to actually add artificial pectin to make a good jam. And since these were berries she'd never used before, she wanted to know if she could figure out how much pectin they had, and if she knew what a closely related berry's pectin content was, would it relate to a nearest relative? And so she asked the question, can pectin content and thus gelling ability be predicted on the basis of berry relatedness? And so what she did was she made jam out of each of these 4 berries, mapped them, their genetic phylogeny here, so you can see how related they are. And she found that pectin content was not related to their genetic ancestry. The 2 that jammed the most were the dogberry and cranberry. So you can see they don't have this pool of liquid forming around them. And those were not the most closely related ones. The dogberry is actually distantly related from all of these other berries. It's related to apples, actually. And so what she found was that you can't infer pectin content based on closely related berries, and you actually have to test each one individually if you want to make jam, which was an interesting result. It wasn't entirely expected either. You might expect berries to have similar levels of pectin. But that's something we've tried not to shy away from on the blog. Having negative results or having results that always make sense at first is something we've tried to address and go over because it's very common in science. And that was really illustrated by another post by Judith Pungar. Judith is actually a neurobiologist, but a fantastic writer. Her posts are very fun to read. She did something called the Great Extravaganza. She started off by talking about her heritage, which was Hungarian, and how Hungarians don't know how to make scrambled eggs well. And she lamented that all of her friends had these delicious scrambled eggs, and she never knew how to make it. And so she wanted to ask the question, how do you make the perfect scrambled eggs? It's something she always wanted to know. She said, turns out for every 10 people you ask, they offer 11 opinions, but what was the best way of doing it? So she had all these variations she wanted to test. So what she did was she decided to focus on the type of ingredients you would add to the eggs. She tested adding heavy cream, 2% water, nothing in butter. And she evaluated them on fluffiness, moisture, and overall taste. And the first thing she found was a big fat negative result. Overall, she very clearly described initially what that perfect scrambled egg was that she wanted to get. And all of these fell short in one way or the other. So none of these variations that people told her would work actually worked. Second of all, she got something that didn't make sense. So if you look at fluffiness, the 3 that produced fluffy eggs were heavy cream, water, and butter. You might expect fat content to be related to how fluffy these eggs would get. And it was related, for example, in butter and heavy cream, but water, which should basically be milk without the fat, didn't produce fluffy eggs. And 2% milk, which should be an intermediate between heavy cream and water, wasn't. It produced heavy eggs. And so this was really hard to make sense out of. And Judith first didn't really write about it, but we asked her to address it. And so she wrote this paragraph in her conclusion slide, which was, but inconclusive results are actually an important part of science, or so my advisor keeps reassuring me. Inconclusive results highlight areas of science that need further exploration. While the magic of scrambled eggs continues to evade me, I know at least that the magic does not lay only in what is added to the eggs. So she took these confusing results and still made sense out of them. She told us that what was happening here wasn't related to either of these things, since they didn't really tell us a clear picture of what was going on. There must be something else going on besides what you add to the eggs to get the perfect scrambled eggs, which is a great way of putting negative results in context. So the last example I want to talk about is, I figured I can't embarrass all of my fellow posters without showing one of my posts. So I've been obsessed for quite some time with hash browns. Made a lot of hash browns. I like breakfast. And so I figured a long time ago that the best way to make hash browns is after shredding the potato on a cheese grater, you need to take those potato shavings and squeeze all of the water out of them that you can. If you don't, you end up with gummy hash browns. They just don't taste right in the middle. They kind of gel together. Now when I did this, I collected the squeezed juices out of the potatoes in shot glasses as shown here. Now I found something really interesting, was that I wasn't just squeezing water out of the potatoes. There's a layer of water here, but at the bottom, there's actually this white layer, potato starch. If you take it out of the shot glass, it's just like oobleck. If any of you are familiar with that, except for potatoes, not corn, which is pretty cool. And, cornstarch is an additive that you use to make gravies more thicker and things. So it could be related to this gumminess that I was tasting with potatoes. So I wanted to know, is it the water or the starches in potatoes that cause them to get their gummy, sticky interior? So I wanted to transform this question into an experiment. And that's rather difficult to do because when you squeeze the potatoes, you get the water and starches out of them. So how do you separate the two effects? I wasn't able to do a perfectly controlled experiment with just water and just starches. So what I did was I did a control where I didn't squeeze anything out. And when I did that, I got my typical gummy potatoes. Didn't taste good at all. I squeezed all the water out, which allowed me to get really crisp, good tasting potatoes as I expected. And then I did one where I squeezed all the water and starches out, and then I added in an equal amount of tap water back into it. So I ended up with a treatment that had the water, but not the starches. And so this third test would allow me to distinguish between these two, hopefully, and whether it was the water or the starches that contributed to it, whether it was gummy or not. And what I found was that it was actually intermediate between the two others. It was slightly gummy, but it wasn't as gummy as the control. So what this allowed me to conclude was that the result, unfortunately, did not fit nicely into either expected answer. In other words, it seems as both water and starches contribute to gumminess. So I was able to put this somewhat confusing result again into context. So, what I wanted to end on was a couple of future directions I'm going to go with this. Basically, one of my jobs at Iridescent, once I start, will be to find ways to make this resource we developed on ScienceFare more useful, to develop useful activities out of it, or lesson plans, or to make more posts that are illustrate things that we want to illustrate. So I'm going to develop a 5-lesson plan after-school science series to pair with a lot of Iridescent's other activities based on the experiments that are done on the blog. I'm going to run a workshop at some point for kids where we teach them how to do a science fair experiment. Then they actually do one, and we help them write it up, and it gets posted on the blog. These are the things I'm going to do for sure. The other things I've been thinking about is something called Ask the Scientist, which I'd love to find someone who might want to partner on this. When I've been thinking about how the scientific method works, the parts I want to focus on are the question asking and the putting the results in context, which are things that you can focus on without doing an experiment. And we're actually looking for more ideas for experiments on the blog. So the idea would be to get a bunch of kids to post questions about what they think are good scientific questions in some way, potentially as a project for class that they would be graded on. We would pick 2 or 3 of those questions and actually do an experiment on them. And then there would be a follow-up activity with the kids where they might evaluate how we did the project. We might test, say whether we took that question and translated it into an experiment well, and what we can do with the results. Did we put them into context well or not? It's just kind of an idea I like to play around with. If anyone's interested in working with me on that, I'd love to talk to you. And also, we have an education section on the blog, and we'd like to expand it, and we can put any sort of thing on there we want. We can transform these into good lesson plans or activities. I'd love to get feedback on what sort of thing would be useful to any of you on that, and any other ideas you might have. So that's all I have. I'd be happy to hear any comments or take questions.
Do you have an experiment? Do you have an experiment you want to do like now and kind of run through what you're talking about the scientific method with it and how you want to run it, how you want to do it?
You mean like experiment on the blog?
Like, give us an example. Right, go for it.
Like not one of the ones I went over? Or?
Yes, like not one of the ones that's already been done, one you just went, like how you would start out and do it, that sort of thing.
A lot of them have started with finding a weird video on the internet or something that really bugged me.
Okay.
I keep thinking of things that were other experiments I wrote on the post because I don't have the inspiration for one I haven't done yet. So like I went home once and my parents told me that I was making chestnuts wrong. That my grandma always put salt and bay leaves in the water to help them peel better. And I said, that's ridiculous, you know. And so I did an experiment where I boiled chestnuts with nothing, with salt, with bay leaves, and with salt and bay leaves, and figured out how easy it was to peel each of those treatments. So the question was, what's the best way to peel bay leaves? I don't know if that really answers your question.
Well, it's just trying to get an idea of how to get the kids to jumpstart them into doing something like that.
Mm-hmm.
them started, like, how do you want to do this? Search duration. You know, like, what would you use? I like the idea of the videos because the kids are always on YouTube or something. Yeah, yeah.
There's tons of cooking blogs that tell you all these different ways.
But I don't think they normally would look at it. But once you say if you say YouTube, you might go, okay.
Yeah, yeah. That's a great way to get inspired. When somebody says something that doesn't seem believable, it's like, well, let's go test it. Sort of our attitude. And actually, on that topic, it's really hard to write a list of rules about how you come up with a good question, I found. And, even that very iterative kind of diagram I saw, it just has sort of high-level concepts. It doesn't actually tell you what you need to do to come up with a good question. And that's something I'm still trying to think through and figure out what sort of advice can be given on that.
I have a question.
Yeah.
You said you have a vendetta against hypothesis.
Sure, we can call it a vendetta.
But then you also mentioned when you were discussing your experiment with the potatoes that you didn't get your expected result, your expected answers. So what's the difference between hypothesis and an expected answer?
It's not that I think they don't exist. Like I had a hypothesis in that clearly. It's just that I think putting the emphasis on hypotheses as an important part of the process is not helpful. It's not something that scientists actually do. And it's also, I think, not very helpful to students. So at some point I ran a summer camp for kids where we guided them through a project they did on marine biology. That's what my degree was on. And I remember we had them come up with questions, and one of them came up with this great question, how fast do different animals flip over? Different kind of slip over, starfish or sea urchins and things. So we're like, okay, now you have to develop your hypothesis. And they said, okay, well, I think the sea urchin will take 5 minutes and the starfish will take 2 minutes. And it was like, well, that kind of fits our definition of a hypothesis, but that didn't help them come up with an experiment. It didn't give them any guidance into the
But isn't it
process.
supposed to be an educated guess, like using prior knowledge to come up with a decision? So like, if they had seen one flip over before, that they thought one would flip over faster than the other, and then like whether or not the experiment proved or disproved that. I mean, it's not just a
Yes.
random guess.
No, it's not supposed to be a random guess. The two things I'll say to that, though, is the one purpose I think hypotheses do have is making you think about the bigger picture, making it an educated guess, realizing how your experiment fits in context. There's no reason that can't just be emphasizing the questioning process. Why do we need the hypothesis itself to emphasize that? I forgot the second thing I was going to say. No, but that is a good point. It is a way to think about what results would you expect and why. How does this fit into a really good context? Yeah. So it's not that they're unuseful. It's just I think they're not nearly as necessary as we make them out to be. Oh, my second point is there are fields that actually just don't use hypotheses. Like in physics experiments, there's often no hypothesis developed when you're testing out quantum mechanics or something like that. You have this elaborate theory for how systems should be predicted. You make measurements on those particles, and they either fit an equation or they don't. If you had to generate a hypothesis, it would be my results fit this equation. But that's obvious, that it's not helpful. Physicists laugh at the idea of hypotheses, whereas biologists fight them to the death. If we're trying to teach a unifying concept of science, we got to make a decision.
So maybe the hypothesis could be more important as a discussion point after the process is finished.
Yeah. I mean, yeah. That would be a great way to use hypotheses. And I'm not going to go on a tirade against that. I think they're great for that sort of thing, figuring out what sort of things to expect.